Hello everyone. Today we are going to discuss about electrophysiology of heart. So what is the purpose of studying the electrophysiology of heart? Let's understand with this short example. Our heart has electromechanical circuit, means there should be electrical impulse for contractility. So here we study how electrical impulse generate, which leads to the contraction of the heart. Let's have a look on this. Our heart's function is to pump the blood by contracting. So how this contraction starts? Any electrical impulse reaches to the muscle or tissue of the heart, which change in the membrane potential or RMP, resting membrane potential, which causes depolarization. The other name we can say that the depolarization equals to contracting. So here is our beautiful heart. Let's zoom it. We have SA node, the AV node, we have bundle office and the Purkinje fibers. So here is our beautiful heart. Let's zoom it. This is SA node, this is AV node. As we know, the SA node has the power of atomicity or the generating impulse by itself. The SA node generate impulse or electrical current which transfers to the AV node. There is some AV nodal delay or the current holding activity occur in the AV node. Then the AV node transfers the current to the bundle of his. Then the bundle of his to the right bundle branches, then to the left bundle branches, which transfers the electrical activity to the Purkinje fibers. Our myocardium or heart muscle have two specialized tissues. There are one is specialized tissue and second one is contractile tissue. The specialized tissue example is the SA node, the AV node or the Purkinje cells. Or example of contractile tissue is atrial myocardium and ventricle myocardium. So let's take the example. We have to take the example of the myocardium. Let's take the one cell of the myocardium and zoom it here. This is the one cardiac myocyte in which we have a nucleus. Nucleus has the property to form a messenger RNA, or we can say that transducer mRNA, which forms the transducer protein, which exchange the sodium potassium ion exchange throughout the membrane. This is the sodium potassium pump. It is works on the phenomena of there is 3 sodium efflux outside and 2 sodium 2 potassium influx inside. As the ion exchange takes place, the more electronegativity generated inside the cells and more electropositive outside the cells. As the gradually sodium efflux outside and the potassium influx inside, it generates electronegativity insides of almost about -5 millivolt, but it is not as much to gain the depolarization. So there is more ion exchange from inside or outside. There is a potassium leaky channels which leak potassium outsides, and also there is sodium insides. As the sodium channels also open, the sodium also move to the outside and potassium also move to the outside. So it forms the electronegativity, more electronegativity insides. One time came when the concentration gradient of outer moving ions or the inward moving ions equal. They are in equilibrium. So it is almost about 90 millivolt. This is called resting membrane potential, RMP, or resting membrane potential, which is about on minus 90 millivolt, where the concentration gradient of the across moving ion are equal. We have discussed the mechanism of how the resting membrane potential generates here. So let's have a look what is resting membrane potential. It is a state of electronegativity which is present in the inner side of cell membrane when the cell is at resting state. Different cells have different resting membrane potentials. So what is the purpose of the resting membrane potential? So as to transmit the signal to other cell membrane, there must be reversal of the polarization, means there is electronegativity inside the cells. So to transfer the signal, the cell membrane generate its polarization to positive sides, which is called action potential. So there are two things which needs to be the generating resting membrane potential. First one is the sodium potassium channel must be there, and second one is the potassium efflux. There must be potassium efflux from outside inside to outside the cell.